Frankfurt, Germany, a city at the heart of Europe and a global hub for commerce, education and technology. And now it's bringing together clinicians, scientists, nurses and many more to share their expertise in bone marrow transplantation and cell therapy, all to help save the lives of patients with blood cancers and other life-threatening diseases. It's the 45th annual meeting of the European Society for Blood and Marrow Transplantation, and we're EBMT TV. Welcome back to the Frankfurt Messe and our third show from the 45th annual meeting of the EBMT. As attendees get to grips with the latest transplantation techniques, best practice guidelines and cutting-edge technology, there's plenty more from us. I'm standing in front of the Hope for Life wall and we'll be finding out about that in just a moment. But also on today's show we'll be hearing from more award winners and also taking a look at the wider team involved in CAR T-cell therapy.
We sequentially used CD19 and CD22 CAR T cells to treat relapsed BALL after HCT.
You need to learn from centres who have delivered a quantity of CAR Ts already, shared experiences and learning from our patients as well.
We need to get out there and collaborate with our clinicians, with our practitioners, because actually pharmacists can be the glue that make this come together really safely for patients.
The EBMT registry is really the tool that we should be using to quality assess and monitor long-term patients receiving CAR Ts.
The new techniques will be available only for a very limited population in very wealthy countries. We have now about 300,000 newborns each year and they are not in these countries, so we have to find solutions for those patients.
We'll be one of the first institutions on Long Island to actually provide CAR T-cell therapy for patients with refractory leukemia and lymphoma.
I'm delighted and honored to receive the John van Rood Award.
I thank very much all the team who works to this study behind me.
We'd like to provide the best overall care for patients from the moment they get here to 10 years or 20 years after they're transplanted.
What we generated is a novel strategy in which we can trace individual stem cells after transplantation.
So work hard, submit your abstract, don't worry about the results. As you work hard, you will be awarded at the end.
Join us for Madrid 2020.
I really enjoyed. This was my first EBMT Congress, but for sure, if it's possible, I will be next year in Madrid.
So Tali, I'm going to be doing some drawing whilst you're telling me a bit about the wall. Can you tell me what is the Hope for Life wall?
This wall was made specially for this conference. I'm coming from Israel and I'm an artist. And what I like here in this conference, that the subject is very hard and heavy and people really express themselves here.
So you've had a good response to it then?
Yeah, people said that they never ever had it, always conference are, coming, hearing, lecture, and then they found something, it was like an experience. I didn't know if people will react, but it was, wow, I think maybe more than 1,000 people. Each one put something small here.
What kind of things have people done on it?
You can see here we love our doctors. Advantage of medicine, they speak about the new medicine and the new treatment that can be for people to be cured.
Well, listen, it's absolutely fantastic. It's obviously had an incredible response. Thank you so much, Tali.
Great.
Well, now I'm back in the EBMT hub, where behind me a couple of special guests are preparing to take a close look at one of this year's top abstracts.
Good morning, Dr. Liu. Thank you for joining me to discuss about your interesting study on the sequential use of CAR versus CD19 and CD22 for relapsed BALL after transplantation. Can you briefly overview the results of your study?
We sequentially used CD19 and CD22 CAR T cells to treat relapsed B-ALL after HCT. Our results showed at the median follow-up time of 10 months among 24 patients, and the overall survival rate was 87.5%, and the MRD negative event-free survival was 58.3%. The median event-free survival time was 12.5 months.
Did you treat pediatric and adult patients in your program?
Yes, we have both pediatric patients and adult patients.
And which were the main complications that were reported in your trial using these two CAR?
The most common toxicity is cytokine release syndrome, and for the patients who relapsed after HCT, another risk is GVHD. So CAR T can induce the GVHD for these patients.
Can you explain us which is the timeline between the first infusion of the CD19 CAR and the second infusion of the CD22?
The time interval between CD19 and CD22 was 1.5 to 6 months. If a patient had a sign of relapse, for example, MRD positive, then we begin with the second CD22 earlier to 1.5 months. If a patient has infection or lower globin or GVHD, then we put off our second CD22 until 6 months.
And do you wait for also hematological recovery, increase in white blood cell and the platelet?
Yes, we also wait. On 2 months after first CAR T, they can recover. But on day 30, usually some patients do not recover.
Can you tell us where are the other disease in which this kind of approach could be applied?
We also do some patients in lymphoma by sequentially apply the CD19, CD22, or even CD20, because lymphoma patients have CD20 expression also on the lymphoma cells.
Thank you very much, and thank you for answering this question.
Thank you.
Great results for the use of CAR T cell therapy in leukemia there. Now, we've been hearing from some of the doctors performing CAR T cell therapy earlier on in the meeting, but what about the wider team? Well, I've been finding out from the nursing, pharmacist and quality management perspective.
The nurses are an integral part in delivering CAR Ts for our patients. We have a wide range of nursing responsibilities, from bedside to when the patient goes home and all the follow-up that needs to ensue.
And how, from the nurse's perspective, has CAR T cell therapy changed, would you say, over the last 12 months?
Well, it's been exponential delivery onto our wards, and the pace of the delivery that we've had to keep up with has been quite challenging.
So what have been the main challenges, would you say?
I would say the pace of the change, the amount of training that we've had to do in a very quick turnaround. Also, managing patients' expectations has been very challenging for us.
So what training is required in the nursing team?
Well, I think learning about the unique toxicities and the side effect profile that CAR Ts have has been quite challenging, and being able to capture all our nurses, including ITU nurses, has been difficult.
What do you think it would take to roll out CAR T therapy to more hospitals and more teams?
I think we need to have, again, really good communication. You need to learn from centres who have delivered a quantity of CAR Ts already, shared experiences, and learning from our patients as well.
Over the last 12 months, what's really happened is, with the advent of the marketed products becoming available, it has suddenly burst into being mainstream. As they say, the CAR-T revolution has arrived, and effectively not just research staff need to know about how to manage CAR-Ts, it needs to be day-to-day work for pharmacists and their teams in hospitals.
How difficult is it to set up the cell therapy from a pharmacist's perspective?
A CAR-T therapy or any cell therapy is a medicine. So fundamentally a pharmacist needs to be aware of putting the patient at the centre of it and making sure that the therapy is optimised and delivered really safely for that patient. But then there's a few additional complexities with cellular therapies because of the nature of the product being a living product. We need to be understanding very carefully about the tracking and tracing of the product, with the product often being autologous for an individual personalised therapy. The consequences of getting this wrong are so huge that we've got to be very, very on top of the operational aspects as well.
What challenges have there been in the administration of the new treatment?
We need to know a lot about cellular medicines. We haven't got that instilled in our education about cells. You know, we come mainly from a chemistry background, and these are biologicals. So we need to get out there and collaborate with our clinicians, with our practitioners, with the commercial sponsors, with the manufacturers, and because actually pharmacists can be the glue that make this come together really safely for patients.
Pharma has quite separate and distinct expectations around how it should be delivered. It doesn't align conveniently with the FACT-JC standards, and I think trying to meet the nuances of their procedures has been tricky from an implementation point of view.
And how have you met them then?
I think most of it's been delivered through on-site inspections, getting their sort of a document packet in advance, having good email communication, contracts, SOPs and so forth. I think is paved the way, and then I think there's also been a lot of chatter amongst the different centres, and I think already there's a shared understanding of what's involved and what kind of lift it takes to get CAR-Ts to open at your site. It's getting your clinical teams familiar with toxicities that they wouldn't necessarily see in an auto, perhaps in an allo setting, getting the pharmacy properly engaged in the management of these advanced therapies on receipt, and the fact that there was now the interplay between pharmacy and the processing lab, or pharmacy and the clinical service. I think that's been a bit of a paradigm shift in how they've had to work.
What kind of quality control and benchmarking can you put in place for a new therapy like this?
Even the sites that are enrolled are really going to have very few patients a year. So I think any kind of statistical analysis at a site level, I think, will not necessarily be very meaningful. I think there's an opportunity to leverage the EBMT registry. Is really the tool that we should be using to quality assess and monitor long-term patients receiving CAR-Ts.
Now, with new cell therapies providing the potential to save the lives of children with rare diseases, I'm here with some top paediatricians in the field to discuss the matter further. Thank you all so much for joining us. Salim, how successful are these treatments?
I think we are at the doorstep of implementing these new techniques for a broad range of diseases. That is not only the genetic diseases like sickle cell disease and thalassemia, but we have also cellular therapy options for leukemias, for AML, in particular currently most advanced for acute lymphoblastic leukemia. But I can imagine that these cellular therapies in the future will be the basis for any form of gene therapy as well, when we are capable of depleting the myeloid niches just with using CAR T cells, for example, or antibodies.
Yosu, what would you say at the moment is the sort of common perception about transplantation?
So there is an understanding that transplantation can be dangerous for these conditions because the rate of complications and mortality is perceived too high. As pediatricians, we often look at these children throughout childhood where there has been a substantial reduction in its mortality and side effects by the medical treatments without looking at the very severe end organ damage. That means that these patients, once they reach the 20s and 30s, have very shortened lifespans. The additional issue is that hemoglobinopathies went into transplantation at a relatively late date, and hence it was thought that only those who had a matched donor could be available, whereas now with the new haploidentical techniques we can offer this to any patient who warrants it.
Marina, how are you able to determine the outcomes for these patients?
The advantage from my point of view of gene therapy upon partially incompatible bone marrow transplantation is that the duration of hospitalization is shortened significantly. The complications linked to the immunosuppression are absent. Conversely, allotransplantation, so complex as performed by José de la Fuente groups, can be performed in very limited places.
Would you like to add something to that?
So I think the most important thing when we look long term about all these therapies is that we need to study how the functional studies are going to pan out. So not just whether we can correct the blood production, but actually whether we're going to make a difference long term, whether there's going to be left impact or damage to the brain or to the lungs. And I think that is what's going to determine which therapy we choose at the end.
The other point that we have to understand is that the new techniques will be available only for a very limited population in very wealthy countries. We have now about 300,000 newborns each year, and they are not in these countries. So we have to find solutions for those patients, and we have outreach programs within the Pediatric Diseases Working Party that clearly show us that we can cure these patients with very limited resources. And I think until gene therapy is widely applicable and demonstrated to be successful, we have to continue with our techniques that we have at the moment, which is stem cell transplantation, either with post-transplant cyclophosphamide or with T-cell depletion, that yield, at least in our hands, excellent results for patients.
I suspect, yes, that in hemoglobinopathies we'll be in the same situation as those who treat leukemia, that we will be able to stratify patients, have a better understanding of the disease, which at the moment is lacking, have a range of therapeutic options, and apply them according to what is best for the patient, working in a multidisciplinary manner, which will include the entire team, not just the transplant physician.
Marina, what's your vision for the future?
For the future, I am very concerned and worried because I don't see how we can bring all this achievement to all the people need to benefit. Gene therapy has now very high cost. In our department, we try, for example, to automatize the process in order to limit the need of very complex and expensive GMP facilities. We try to reduce the time for the procedures and also to develop a drug for conditioning the patient without using chemotherapy.
Well, it's a debate we could continue to talk about for a long time, but we'll leave it there. Thank you all very much for joining us.
Thank you to you.
Well, let's go over to the United States now, where the Northwell Health Cancer Institute is one of a select number of FACT-approved cellular therapy programs, delivering the very latest therapies to patients.
What's really unique about the Northwell Health Cancer Institute is that we're part of an integrated healthcare system which allows us to provide the entire range and scope of services. So we can care from patients through their entire journey, from screening and diagnosis all the way through treatment and ultimately onto survivorship, where they're followed for the rest of their lives. Our goal is to really provide comprehensive cancer care. Our standard of care should be to provide comprehensive medical care. We want to provide patients with all aspects of their treatment. We want to avail them of the latest and best screening procedures, diagnostic tests. We want to dramatically expand our portfolio of cancer clinical trials to bring the latest cutting-edge treatments to patients. We are extremely proud of our incredible stem cell and cellular therapy programs. One of the things that we're most excited about is we'll be one of the first institutions on Long Island to actually provide CAR T-cell therapy for patients with refractory leukemia and lymphoma.
As the director of the stem cell transplant program, I oversee all aspects of the process. So we do offer our patients a continuum of care where we are collecting the cells, we are processing the cells, and we are also doing the transplants. Patients with relapsed lymphoma or with leukemia, they are able to get their initial treatments and then move on to the process of stem cell transplantation.
The role of the Northwell Health Adult HPC Transplant Program in the health system is that it's the only program in the health system that offers stem cell transplant to our patients. This is a vital service that is offered to patients who live in Brooklyn, Queens, and Long Island. And there's 3 components to the program. There is the pre-transplant workup that happens at the Monte Cancer Center. There's the inpatient stay where the patient actually receives their stem cell transplant, and then there's outpatient follow-up back at the Monte Cancer Center after the patient's discharged.
Every day it is a gift to have the opportunity to care for patients who are in their most vulnerable times, to be able to be a part of the continuum of care for these patients, and to be able to provide expertise and specialized knowledge and treatment that can offer the best chance for cure for these patients.
Some of the strengths of our program is that it's at a very individualized point of care for our patients. We do everything we can to make the patients know that we care about them. We know a lot about them. We know about their birthdays. We know about their children's names. We do everything we can to personalize the care.
And we really function almost like an extended family, and our patients then become part of that family, and our extended family is ever growing.
My name is Greg Holgerson. I'm 45 years old. I live in Bohemia, New York, and I'm married to my wife, Christine, and I have 2 kids, my daughter Fiona, who's 9, and my son JT, who's 6. I'm currently a police officer with the Nassau County Police Department, and I've been with them coming up on 19 years. When I first got diagnosed, due to my age and health, they decided to try and treat me with chemotherapy over the course of a year. So about every 2 months I came in for 5 days of treatment. I had gone back to work, everything was fine. I went in for one more bone marrow test, and that's when I got the phone call that I had come back. I knew that I had to have the bone marrow transplant. Out of the blue, they found a donor on a registry in England that was a perfect match, perfect health and everything, and that's how it started. All I knew that it was a female and that she was from England. I knew nothing else. Through the 2 years, we exchanged cards, but when I finally got permission to, we then got her email address and phone number, and then we started talking all the time. Then when the Monte Center and everybody brought her over to finally meet her in person, it was unbelievable. She's an awesome woman. So, I'm 5 years post-transplant. Overall, my health is good, and I just take it one day at a time.
The Don Monte Memorial Research Foundation has supported the stem cell transplant program from the beginning, and I do not believe that we would be as successful as we are without the help of Caroline Monte Saladino.
Caroline Monte Saladino and her family have decades-long commitment to caring for our patients and caring for the staff. They took the tragedy of their son dying at a very young age and turned it into something very positive. Caroline's parents had a legacy that they've handed on to her where they wanted to have a human touch along with the cancer diagnosis. Caroline rounds on our patients and meets with them, speaks with them. She supports our programs for our patients, which is quarterly parties that we have where we celebrate St. Patrick's Day, and Christmas, and 4th of July, and Halloween, so that patients know there is life beyond these walls here, that there are things to look forward to.
The Northwell Health Cancer Institute is not a single facility. It's a system of 23 hospitals. We need to make sure that in the hospitals that are providing cancer care, which many of them do, that we're standardizing that care, that we're providing the best care possible in all of those locations, and that we're also availing patients of the latest cutting-edge clinical trials in all of those facilities.
I am a believer in transplant democracy. I believe that we need to provide equal access and outcomes for all. And at the end of the day, I do believe that everybody receives a beautiful reward.
In our center we are treating babies, toddlers, school children up to young adults. Excellent patient care for pediatric patients could only be guaranteed in dedicated and specialized pediatric centers.
We'd like to provide the best overall care for patients from the moment they get here to 10 years or 20 years after they're transplanted.
We'll be one of the first institutions on Long Island to actually provide CAR T-cell therapy for patients with refractory leukemia and lymphoma.
Physicians in the area around get notice of us, and they are presenting their patients to us because they believe in us to do our good job. That is what makes us proud.
What makes the treatment unique in our department is the combination of high-tech medicine, but not forgetting about the supportive care of the whole family.
Theracos ECP immunomodulation employs the patient's own immune cells to modulate dysregulated immune function. For more than 30 years, Theracos has been pioneering the science of immunomodulation through ECP and its therapeutic applications.
Well, we've heard from the winners of the Van Beckham and the Basic Science Awards at EBMT 2019. But the awards don't stop there. We've been catching up with a couple more of the best and brightest.
We published a paper in Science Translational Medicine describing how the human thymopoiesis is influenced by a new genetic variation within in the locus the TCR alpha and TCR delta, the variability of thymic function in a large-scale study. And our population knowledge approach revealed new influence by polymorphism in the thymic function in healthy adults, and it is potentially a broad implication in the precision medicine. I'm delighted and honored and received the John Van Rood Award, and obtain this award. It's a supportive and encouraging for me to continue my scientific research, and it was also a great validation of the work done by our talented research team. The EBMT annual meeting, it's a unique opportunity to meet with specialists in the field and to share experiences and develop cooperative studies.
A study about patients with Hodgkin lymphoma with relapsed and refractory disease who underwent the haploidentical transplantation with prior checkpoint inhibition. So we want to compare if the outcomes were different between patients who received checkpoint inhibitor treatment before haploidentical transplantation and patients who received haploidentical transplantation without prior checkpoint inhibition. We find that patients who received transplant with prior checkpoint inhibition had a higher PFS and a lower relapse rate compared with patients who received transplant without prior checkpoint inhibition. I thank very much all the team who works to this study behind me, so all my colleagues from the three European centers who enrolled the patients and helped me to write and design this study.
New research well recognised there by the EBMT. Next, with more in the way of novel treatments, from cell therapy to immunotherapy, let's head over to the Siteman Cancer Center, based in Missouri in the United States.
We're probably the world's leading center for cancer genomics, having sequenced the first cancer genome in the world in 2008, and that's been really the start of personalized medicine in the world. The basic research that we do now is the fuel that lights the fire of clinical research in a year or two. And so we have a large number of basic science investigators that are doing very fundamental things. We've been very interested in the fundamental aspects of graft versus host disease, and I think we've been the center that started to really look at a pathway called JAK signaling pathway for mitigation or treatment of graft versus host disease. Probably one of the most frequently used drugs for the treatment of graft versus host disease are JAK inhibitors, and I think it's based on our basic science laboratory discoveries here.
So one rule that John DiPerzio and I have had for the essence of the studies that have come from this group is that we start at the bedside. We let the patients define the rules. That is an essential feature of all of our work. We do not model things based usually on hypothesis testing in the laboratory, trying to take it towards the patients. We try to make the observations using patient material first. For a long time, we've been interested in the molecular underpinnings of acute myeloid leukemia and its related disease, myelodysplastic syndromes. When the human genome was sequenced back around 2000, we were very interested in utilizing that information to really begin to better understand the mutations that caused AML. The toolbox that we have to work with this kind of data is getting better and better. Integrating all that material together in a clinically useful way is very important. It's also important to do it not ad hoc but prospectively, using well-designed clinical trials that are designed to determine whether these testing ideas that we're working on now really make a difference in terms of how the patients do. We have to not just use the data because it's there, but we have to use it in a way that is instructive, informative, that clearly, by the best scientific criteria, tell us that we're making a difference in patients' lives, improving their outcomes because of the testing that we're doing.
Well, one of the things that I've always liked about Washington University is the close interaction between the basic scientists, the clinicians, and the patients. So what's really heartwarming as a scientist is to see the bench to bedside. And so we do a really nice job of facilitating interaction between our basic scientists and our clinicians. And I think it really helps us translate this into better treatment. One of the things I do is I'm the head of the SPORE in Leukemia program. Washington University has two very large multi-center grants. One is the Genomics of AML PPG, that's led by Tim Lay, and then the SPORE in Leukemia. So together these two large grants bring about $26 million of funding in from the NCI and involve about 30 faculty. Todd Feniger here is leading an effort to develop a new type of immunotherapy using natural killer cells. It's early days, but there's some very exciting results. And so this is potentially something that he's used right now mostly for leukemia, but he has plans to use this in lymphoma, myeloma, and who knows, perhaps other solid tumors too.
I'm more on the clinical and translational side of research and mainly focused on cellular therapies. Cellular therapy mainly using cells to treat cancer or cure cancers. That include chimeric antigen receptor T-cell therapy or CAR T-cell therapy. With the chimeric antigen receptor T-cell therapy, the chance of going to complete remission is more than 50%. So that's a dramatic improvement. And now we have that as a standard of care. That's this bench to bedside collaboration between scientists and physicians and clinicians going back and forth is absolutely necessary to make this treatment more effective and cure more patients.
We'd like to provide the best overall care for patients from soup to nuts, from the moment they get here to 10 years or 20 years after they're transplanted. I always feel relieved that the patient has come to see us. because I feel like the patient's in the right place. I would say that we'll provide as good a care as anyone can provide the patient.
This year, the EBMT has launched a new research grant in collaboration with the Department of Experimental and Clinical Medicine at the University of Florence. I'm delighted now to be with the recipient of that grant. Miriam, thank you so much for joining us.
Thank you.
Tell me first of all, why you've won this grant.
The work I've been doing is about stem cell tracing. And I think we're using a very innovative approach to revisit long-standing questions in stem cell transplant medicine. Which probably is one of the reasons for getting this award.
So what sort of questions are you answering?
Well, one of the things I'm amazed by is how such a small bag of stem cells can actually regenerate a full blood system and do so throughout the life of a person. Like, I'm a pediatrician, so in our field, our stem cell transplant recipients will need to survive for many, many decades with these new stem cells. And my question in my research is, how do these stem cells do that?
And why is the work that you're doing so important?
One of the major focuses of us as transplant physicians would be to maintain health for prolonged periods of time and to actually give children the optimal treatment. And one of the focuses of my research is actually to see how transplanted cells make blood, but also how the transplant procedure affects the integrity of the stem cells and how we can actually maintain that and improve that.
So what did you find? How can you?
Well, what we generated is a novel strategy in which we can trace individual stem cells after transplantation. We've used this strategy thus far to study normal aging, and in this field, or in this application, we're using this same strategy to trace stem cells after transplantation.
What do you think that the awarding of this grant will help you to achieve?
I entered this field a couple of years ago, and this is really my first grant in stem cell transplantation. So it's really a recognition that the ideas and the projects that we're pursuing are actually recognized by the field. For us, it's really an incentive to pursue this project and to make it even bigger. There's millions of questions that you could address with this award, and this is just the first step of, I hope, many.
Miriam, many congratulations and thank you for joining us.
Well, thank you.
Well, we heard from some of the EBMT's young ambassadors yesterday, but that's not the only way that the EBMT is encouraging young researchers. We got the chance to meet some of the best young abstract awards winners. Let's hear from them.
My poster is about T-cell therapies against viral reactivations, in particular against EBV.
My presentation is about the possibility of being able to make our own academic CAR T-cell products.
It's a presentation of our results of a new protocol of haploidentical transplantation in pediatric patients with high-risk acute leukemia.
Our presentation was about post-transplant prediction of outcome in myelofibrosis, primary and secondary to essential thrombocythemia and polycythemia vera.
So the topic was how IL-7 and IL-15, which are homeostatic cytokines, impact the T-cell reconstitution after transplantation in 80 children transplanted for acute leukemia.
My poster was about treselfan-based conditioning and the allogeneic hematopoietic stem cell transplantation of pediatric non-malignant disease.
I was really happy when I heard that my poster had been accepted for an award. It was something that I think both for me and for the group was really nice.
I am very, very happy. It's beautiful work with little patients.
It will increase awareness of our study, which I think is very interesting, and hopefully also help us start collaboration later on to expand some of the interesting findings.
For me, it's an opportunity to get to know other young people who are really in a new field like CAR-T, etc., and to learn from them.
This gives me a push to continue to do more research and to establish my unit, so I returned back to my country. I'm from Egypt.
I would say to other people that are thinking of submitting abstracts to honestly go ahead and do it. It's a chance to share the research that you're doing, to see how much more people in the field are working on similar subjects.
Just do it and take the risk, and don't be nervous if you have to face difficult questions. Try to answer them as honestly as you can.
So work hard, submit your abstract, don't worry about the results. As you work hard, you will be awarded at the end.
Definitely do it, because it's very rewarding also to go to the EBMT.
Now, as EBMT 2019 starts to wind down, it's time to look ahead to next year as EBMT heads to Madrid.
As a president of the 46th edition of the annual meeting of the EBMT,
and as the scientific chair,
we would like to invite you to join us next year in Madrid.
EBMT 2020 will give us the possibility to discuss for the first time real-world data on CAR-T cells in European centers.
This meeting will be a superb platform for a scientific, educational and networking exchange. Join us for Madrid 2020.
Really exciting news there, and we cannot wait for next year. But of course, EBMT 2019 isn't over yet. We wanted to find out from you what your meeting highlights have been and just what keeps you coming back.
I really like the EBMT because it talks about a lot of things, about hematological transplant, and yeah.
And maybe we can meet another people from another countries that they are doing the transplants, but in another way, and now we make another friend.
We have made a lot of friends today.
It's a very important meeting in the field for transplantation, and we have a chance to have a very good idea of development of transplantation in not only in Europe, mainly in Europe, but also worldwide.
Yeah, good topics were included in that, and it's comfortable environment for us also. As we are from India, from another country, it's a new and first time we are coming here.
So I work with clinical research, so I have a bunch of investigators here. So I meet with some of them, and for me it's really rewarding work.
I really enjoyed. This was my first EBMT Congress, but for sure, if it's possible, I will be next year in Madrid.
Well, that brings us to the end of today's programme, and in fact that's all from EBMT TV for this year. And what a fantastic meeting it's been. We've heard more about CAR-T cell therapy, looked into survivorship training for nurses, and heard insights from transplant coordinators, and much, much more. Don't forget that you can keep watching via the EBMT app, and you can keep sharing there as well, so you and your colleagues don't need to miss a thing. So from all of us here at EBMT TV, we'll see you in Madrid 2020.